Welcome, everybody. My name is Anthony Austin. I am the Global Marketing Manager here at Medix Biochemica. Today, our webinar is entitled Empowering Global Health, Rapid Diagnostics and Infectious Disease Detection from Development to Implementation. We're so excited that you're here today. Before we get into some of the more exciting stuff, just a bit of housekeeping on my part. First off, you'll notice that the chat function is disabled for today. However, the Q&A button is open, and we encourage you to submit any questions you have during the webinar for us. We will hold a Q&A session at the end of this webinar. However, please be aware that we might run out of time, or there might be a particularly difficult question that we need to do a little bit of research on our side to get you the answers that you need. But in any case, we will follow up with you through email after the webinar is over and get you the answers to all the questions that were asked during the webinar and those that we weren't able to get to. You'll notice as you joined, the webinar is also being recorded, and that's so we can provide you with an on-demand link. We will also provide you with the slides. Please feel free to share with any friends, colleagues, neighbors, anybody who might be interested in this topic. And it's also especially for those who were able to register but weren't able to attend today. Welcome, of course, to everybody. If you're not familiar with Medix Biochemica, we are an independent, international, and industry-leading raw material supplier, especially for the in vitro diagnostics market. We do have experts within our company across many different applications, including immunoassays, clinical chemistry, enzymes, molecular diagnostic reagents, biospecimens, base matrices, a number of different related fields. This year, we're excited to be celebrating our 40th year in existence. So in 1985, we started out in Finland, where we were one of the first and key players on the field to provide at scale and at quality monoclonal antibodies for test manufacturers. Since then, and especially in the past 5 or 6 years, we've really expanded globally, well, both geographically, but as well as the product portfolio. So now we have manufacturing sites not only in Finland, where we started, but also in Germany, France, the United States, as well as in China. And we've also expanded our portfolio product-wise to not only include the same monoclonal antibodies that we did in 1985, but many more of them, as well as antigens, proteins, enzymes, biospecimens, base matrices, molecular diagnostic reagents, a number of related critical raw materials that are essential for test manufacturers. As recently as January of this year, we've also included immunoassay stabilizers and blockers to that portfolio. And it's really our vision as a company to be your first choice raw material supplier. And we really hope that we can work with you to understand your needs, your pain points, and how we can help you build quality in each and every one of your tests. Today, we're excited to be joined by Richard Campbell. He's a founder and managing director at Lateral Dx. And he'll spend some time walking through some slides that highlights the assay development process, as well as where you should really focus and spend some time and effort so that you can ensure success in this process and make it as efficient as possible. After that, we'll pass it over to Amy Moore. She's the global product manager here at Medix Biochemica and is in charge of the infectious disease portfolio. And she'll take you through some stats that highlight the importance of infectious disease testing, as well as give you a sneak peek into some of the solutions that Medix Biochemica can provide. So with that, Richard, I will stop sharing my screen. I'll turn my video and my audio off. Thank you all I'll also end the poll. Thank you all for your feedback there. So if you just give me 1 second, I will stop sharing and click all the buttons, and then it's over to you.
Okay, hopefully we're all loaded up here now. And as Anthony said, I will be giving you a kind of understanding of the lateral flow test development process and picking out some key parts in that process that we think over our years of experience are worth paying attention to. So Lateral Dx, we were established in 2019. We've been going 6 years now, but we come with a long history of developing lateral flow tests. Myself, I've been doing it for 25, 30 years, and a lot of our scientists and staff have similar levels of experience. We focus on lateral flow. That's what we do, and that's why we consider ourselves the experts in this field. And we're really looking for that customer-focused approach to contract development and contract manufacture of lateral flow tests. We are operating in 4 main areas. We do the contract development. We take things from proof of concept through feasibility. We also do contract manufacture of products. And we have a consultancy service for people who are getting started in the industry or looking for specific advice on scale up or how to approach certain problems. And I'll talk a little bit about our rapid antibody screening as part of the slides, but it's our process for finding those, really good antibodies and determining which ones to include in your lateral flow test. So Lateral Dx has lots of experience of developing lateral flows. We've covered many different nanoparticle types and conjugated the most of those, conjugated antibodies and other binders to those. Worked in a lot of different sample matrices, from your usual blood, serum and plasma, down to more kind of esoteric sample matrices such as milk and fuel and soil and things like that. Obviously, you'll expect us to have worked in lots of different formats, from antibody sandwiches to serological to competitive test formats. And we're now doing a lot more of our developments, which are fully quantitative, as well as the more standard visually read qualitative or semi-quantitative test formats. Today's webinar is around infectious disease, which obviously we have done lots of work in, COVID-19, flu, other infectious diseases such as malaria and HIV. But we also operate across many other varied aspects and sectors and really where point of care can be applied the best, where you're looking for that fast on-site solution to the problem. And then, we also look at and use lots of different types of binders, the usual antibodies, poly, mono, and more commonly these days recombinant, but have also dealt with synthetic binders such as affimers and aptamers. So hopefully that gives a little flavor of what Lateral Dx do and what our experience is. I'll now move into the kind of process of how do you develop that lateral flow test for your infectious disease application. So when we're approaching it as a contract development company, what we really do at the beginning is determine those user needs and what those key design inputs are going to be for the project. It's worth spending time on those, getting them right, and making sure that you get the development off to a good start. You can have some fuzziness in those inputs in the beginning, especially if you're maybe starting in the proof of concept stage. But I would really advise to try and get those nailed down as well as you can at the beginning of the process. And that probably involves speaking to or working out what the customer needs, what the particular application is, and what those critical parameters will be in that final test. Is it sensitivity? Is it specificity? Is it speed to result? Is it quantitation? Lots of other points. So once we've got that, we then produce a project plan and a timeline comes out of that, and we start the work, entering into the feasibility stage. And our aim here is to get to a feasibility prototype and deliver a feasibility report. So we're really testing out different formats of how the test might be put together. We're screening different reagents. We're testing prototypes as we go along. And we'll probably set up an initial R&D stability at this point to give us an idea of the overall stability of the test. Then we'll have a phase-gated review of the project in conjunction with the customer. And if everybody's in agreement and we're meeting the requirements, we move then into optimization. Optimization can be a very overlooked process. And it can be difficult for us to show the value of that to customers. But it really is important for that raw material robustness and for that scale-up processing. We're then moving into transfer and validation, and I'll come back to that in the talk later in the presentation, and cover some of those transfer and validation activities. And then all going well, you're then moving into routine manufacture and start producing your batch manufacturing records and any trending of the product. So whilst in feasibility, there are some key considerations, technical ones that you should be thinking about. And I'll go through a few of these in the following slides. So initially, you have to determine what format the assay is going to take, or you might actually want to evaluate both kind of main formats. Larger analytes, you can use that sandwich format, where you have 2 binders, one on your test line and one on your signal particle. That works for things like proteins and bacteria and most viruses. Then again, if you're detecting a smaller analyte, you might need to use a competitive format where there's only a single binding site for your binder, your antibody, and that's more for things like drugs and toxins and maybe pharmaceuticals and things like that. And then each of those has a different output. The sandwich format has a more understandable output where the test line develops with a positive, and the competitive, that's the absence of the signal that indicates the positive. There's also a diagram here showing you the main components of the lateral flow test, and we'll go into those in a little bit more detail as we move through. Big choice to make is what is your label that you're going to be binding these high quality reagents to. The industry standard is still colloidal gold, and we would always start our developments using colloidal gold, and then we add the other particles as and when they're needed. You might, for example, need dyed microspheres if you're doing multiplexing and have require multiple colors. You might use a fluorescent nanoparticle if you're requiring increases in sensitivity. We've also had a lot of success of using cellulose nanobeads to not only provide additional colors, but also to increase the sensitivity in the lateral flow tests we're developing. So a little bit more on gold colloid. They come in a range of sizes from 20 to 60. Some 80 nanometers are also available, but generally most people are using a 40 nanometer for use in lateral flow, giving you the right balance of signal and binding capacity, and also flow through the lateral flow test. The conjugation is happening through electrostatic forces. We're binding the antibodies via sulfur-containing amino acids in your antibody. Optimization of that conjugate process is really important, and we're adjusting pH and also the buffer that we're using for the binding, and we're then stabilizing with different agents, but usually starting with BSA. And then we have, that gold colloid is, as I've said, that industry standard for use in lateral flows still to this day. I've picked out fluorescent nanoparticles, which are becoming more and more popular. Majority of these are using the europium nanoparticle. These sit in the range of about 100 to 300 nanometers, so bigger than the gold colloid, and that should be taken into consideration when you're thinking about flow through the membrane. Usually this conjugation is using EDC, NHS, covalent coupling methods, but there are other methods for conjugating to the fluorescent nanoparticles. And people are choosing these usually when they need that boost in sensitivity. And the europium-based particles give you that large Stokes shift that allows you to excite the particle and then measure a wavelength that's reasonably far away from that, preventing any kind of interference from the excitation wavelength. Okay, so now we're into the different components that go into the lateral flow test. I'll pick out a few points on all these, but won't go into all the detail, but happy to answer any questions on some of the materials and their functions after this. So the sample pad does a lot of the heavy lifting in the lateral flow test. So if you're dealing with a finger prick sample, we have to separate the red blood cells from the plasma. So we need something in that sample pad that's going to retain the red blood cells. We're usually filtering the sample in some way, removing some of the viscous elements from it or removing components that we don't want to run up the lateral flow strip. Lateral flows don't have wash steps, so removal of things that you don't want interacting further up the test can be a good strategy to get good performance. And in the kind of area of infectious diseases, it can be really important to lyse the virus or bacteria or infectious agent. Not only does that make it, it kills it, but it also increases the amount of available antigen for the lateral flow test. So instead of detecting one virus, you can mash it up, lyse it, and there'll be multiple epitopes, multiple antigens that can all take binding events. And a lot of the COVID-19 tests obviously did that to improve their sensitivity. Then we have the conjugate pad where we're putting our detector, our gold colloid, or our fluorescent particle. We want that to take the conjugate well and manufacture and then release it when liquid is applied from the sample. The nitrocellulose membrane controls a lot of the aspects of the test, sensitivity, the speed of the test, and we want it to bind the test and the control line reagents with good affinity and give us reduced levels of non-specific binding if possible. The absorption wick doesn't really draw the sample along the strip, but it does hold the sample. And so we have to calculate the sample volume that's being applied and then know what we're going to have in terms of absorption capability. The backing cards providing the structure to the test, and the buffer that we're running through the test allows us to change the environment in which the lateral flow test is performing, allowing us to favor binding of different antigens to the antibodies in the system. These balances in the lateral flow test are usually working in an inverse relationship. And so as we improve sensitivity, we might be losing specificity. And it's important that when you're looking at what your test is going to be used for, that you look at that intended use for the test by whoever's going to run it. Is it a screening test where you want to minimize missed cases? Is it a confirmatory test where you want to minimize false positives? So really think about what you want the test to do, and then you let your developer, or when you're developing yourself, you bear that in mind and skew the test to meet the characteristics that you desire. We spend most of our time in Lateral Dx removing non-specific binding from lateral flow test. As I've said, there is no wash step, so everything is happening all at once. So there are unwanted interactions between the assay components, antibodies, and the target analytes and other components in the lateral flow test. And we want to reduce those to minimize false positives in the test or eliminate them. Usually caused by hydrophobic interactions and can come from matrix effects from complex sample types such as blood and nasal swabs and such like. It can be minimized through selecting the right antibody, and this is the main way we use for avoiding non-specific binding, is to find that quality antibody pair that elicits very low or no non-specific binding. your matrix. But we can apply other techniques, such as using surfactants and salts to reduce that binding. So as I've said, that antibody selection part is really important, and you should consider, are you using a commercial source or are you developing it in-house? If you're developing in-house, do you have the quality manufacturing in order to produce the same thing again and again? If you're using commercial, has it got the data behind it that you need for your application? Will it work in your format? And then once you're thinking about that commercial source, what is the pricing? Is it suitable for lateral flow applications? How available is it? Does it need to be manufactured? Is it on the shelf? Some antibody providers will charge you a license fee, so definitely check that that is not the case, or if it is, then find out what that licensing fee might be. Choose your antibody, mono, poly, or recombinant, and you can choose that antibody based on your application, and polyclonals work very well, especially when you're maybe trying to detect a wide range of antigens, maybe many subgroups of a bacteria or other sort of variability in your sample. How many should you screen? How to perform that screening? Should you just pick the recommended pairings from the manufacturer, or should you do a full matrix? I'll talk a little bit more about that next. A lot of people do an ELISA pre-screen. We've now stopped using ELISA pre-screening because it really isn't giving us the data that we wanted when we then moved it forward into lateral flow. We now do all the screening in that lateral flow format. And then when you're doing the screening, are you screening in buffer, the actual matrix, so plasma, or are you using characterized samples to do that screening process? So as I said, at Lateral Dx, we have developed what we call a rapid antibody screening for lateral flow system, where we take a range of antibodies. In this case, it's 5 antibodies. We deposit them all, we conjugate them all to the signal particle, and that can be any particle, fluorescent, gold, carbon, whatever. We then deposit them all on the nitrocellulose, and we test all combinations. So 5 antibodies gives you a total of 25 combinations. And the little graph here, the red bars show the positive and the black show the negative. And we're looking for the ones that give us the most signal with the least amount of negative, so kind of the differential. And then we can take the best of those forward and do a crude limit of detection to give us a rough idea of the sensitivity of each of these pairings going forward. And that's what we then use to take forward into the full development. When you're developing, what are you using in order to make your decisions? So what are your development samples? Should you be using purified or maybe a recombinant analyte or protein? These days, the majority probably are recombinant. It can be more and more difficult to get those purified proteins direct from the host or from the sample. A lot of people will start in spike buffer or spike matrix, and then you've got to make a decision of what is a good number of samples to have in your sample panel during the development process. Where are you sourcing them from, and how much characterization do they have? I'm getting close on my time, so I'm going to move relatively fast through these next stages. So optimization, as you can see on the screen here, we're looking at all the components that go into the lateral flow test and looking at, the manufacturer, where are they coming from, what's the variability on them, what are the tolerances on all the measurements, how available are the materials. For the strip dimensions, we're working out overlaps and the tolerances of all those overlaps. And then how does that interact with the housing and the pinch points and the closure? And then all of this is with an eye to how manufacturable it is. can you scale this up? Can you make it in volume? How efficient is that process going to be? So as I said at the beginning, it's a really important process and can be overlooked. Once people get a feasibility prototype, they think the job's done, but actually the hard work usually starts in the optimization phase. There's lots of points that need tackled. In the optimization, we will also be developing the QC panel, which would be used in manufacture for determining batch-to-batch variability and making sure the test meets its critical performance characteristics. We're looking at where the test is made, the facilities and the utilities. We're usually making a pilot batch and having that evaluated. And then we're doing another informal stability trial, finishing the packaging, getting all that done. And all that ties in with the manufacturing paperwork and the validation master plan, which gathers all that information together. And then lastly, we're into the design, verification, and validation, where we're setting QC specifications. We're working on the device history record, the device manufacturing record, and doing method validation, all the way through to producing 3 validation batches at scale, making sure they pass specification, putting the test on formal stability to get that data. And what you're looking for here in this design and verification and validation phase is no surprises. You've hopefully done most of the work in feasibility and optimization. And here you're just generating data for claims and performance and making sure you have a robust product. So that's me finishing up here, but thank you very much. And I will now pass back to Anthony. Thank you.
Thanks, Richard. And Amy, I think you're up.
Hi, everyone. I'm Amy Moore, the Global Product Manager for Infectious Disease at Medix Biochemica. And thank you, Richard, for your presentation of very valuable information. As we know, we're facing a lot of health crises in the world today, including infectious disease. And one of the concerns is that we have an aging population. And the World Health Organization estimates that the number of people aged 60 or over will double by the year 2050. And as we know, some infectious diseases disproportionately affect older people. So that's a factor in our planning. And also people have more vaccine hesitancy. And what this is bringing about are different vaccine preventable outbreaks, such as measles that is happening in the world right now, Bordetella pertussis or whooping cough. Another factor is increasing globalization and travel. So any isolated infection in one area of the world can spread very easily, either through airplane travel, migration of people, trading of goods that may contain infectious disease, contact with animals. So things that may have been centered in one part of the world can be spread easily anywhere. Another factor that goes along with that a bit is climate change. And the world is experiencing increasing temperatures. And what this does is tends to cause more unexpected weather patterns, more unpredictable and greater events, storms, which would include flooding, which would increase the chances of infectious disease for gastrointestinal infections, respiratory infections, and also increase the habitat of vectors such as mosquitoes. And there are many places in the world that were never able to acquire a natural infection from some of these vector-borne diseases. But now that the temperatures are a little bit warmer in some areas, the insects are able to have a very nice habitat for themselves. And maybe the most pressing issue is antibiotic resistance, where for too long we've been prescribing antibiotics for viral infections, and it's not effective. And what it is doing is creating bacterial strains that are resistant to very common antibiotics and are much more difficult to treat. Those infections are much more difficult to treat now. We have some options for diagnostic testing for infectious diseases over the years, and the gold standard has always been culture, but this can take quite a bit of time. It's very slow test results, not the best sensitivity, can be high cost, and really requires much labor to produce results. Molecular testing, which has great sensitivity and specificity, but has higher costs, possibly reimbursement issues, and often requires specially trained lab personnel and equipment. Additionally, there's serology testing where detection of the patient's antibody is performed. And this is typically simple. It can be rapid. And IgM antibodies indicate a more recent infection, and IgG positive results indicate a later diagnosis or possibly a previous exposure. A disadvantage for serology assays is that they are very limited in the information they tell you about what the patient is experiencing right now. So not much utility for acute infections. And antigen detection is probably what we're most familiar with COVID testing. And this can provide direct evidence of an acute infection. So that person is infected right now with very fast test results, a high positive predicted value. So if you're positive, there's high likelihood that you truly are positive and are infected with that agent. Typically simple to use and of a lower cost. The one drawback is that they tend to have a lower sensitivity. And all except culture can be considered or can be engineered to be rapid diagnostic tests that can be used at the point of care. So at the patient, right at the patient's location. And the World Health Organization has established some criteria for the ideal point of care test. And they should be affordable. They should be specific and sensitive. They should be user-friendly. They should be rapid and robust. They should be equipment-free and deliverable. And one type of test that really hits these marks are lateral flow. And some other benefits and advantages to point of care testing and rapid point of care testing is, number one, the healthcare provider is able to make timely decisions. So this would be possibly isolating the patient before they leave the office, providing appropriate care whether that's supportive care or medication, which could include antibiotics, and also provide additional testing. One type of infectious agent may lead you to additional testing that would help the community and the patient, and also patient education for how to manage this infectious disease. For outbreak situations, these types of assays can be brought to the site of the outbreak and can be used to track spread of the infectious agent, can identify any high-risk areas that may need special attention by healthcare workers or other authorities, can also help to decentralize testing that is happening in the area so that the hospitals do not become overwhelmed with all of the testing needs. And it may also help to slow or prevent community transmission. And for any resource limited areas, and this would include any area that's experiencing a natural disaster where the power may be out or facilities are not available, something that does not require equipment or refrigeration is key. In lower middle income countries or in rural areas, this would also be key, where you don't have to transport equipment. And these tests can also be beneficial to reduce antimicrobial resistance by providing an accurate diagnosis so that antibiotics that are not unnecessarily prescribed. And lastly, patients are really demanding more self-testing. I think we've all become accustomed to COVID testing, and we want our answers quickly and preferably at home. And we're really demanding that. And a lot of that is for the convenience, so that there's no medical appointments, we don't need to go anywhere, and also for privacy. One key point that needs to be considered is that there's no real linkage to care by a medical officer. So this is a point that that we need to work on. So Medix Biochemica's product selection is very vast, and we have a lot of solutions for all of these infectious disease problems. And currently we have over 1,000 products that are composed of monoclonal antibodies, polyclonal antibodies, and recombinant and native antigens. And one of our strengths are respiratory products, but we also, as you can see on the graph on the left, have a wide variety of subclinical areas supported. So other than respiratory, there's sexually transmitted infections, gastrointestinal infections, water and foodborne, vector-borne disease, hepatitis B, C, and other hepatitis viruses, and also veterinary infectious disease. So with our strength being respiratory products, I wanted to mention acute respiratory infections first. And certainly with the COVID pandemic and flu season just wrapping up, we really can't discount the severity of respiratory infectious diseases. And acute respiratory infections are one of the leading causes of mortality worldwide. And the burden for that mortality falls really to the very young and to the elderly. And in many cases, these infectious diseases have very similar symptoms. So diagnosing based on symptoms is challenging. And also the respiratory infections are usually the top cause of unnecessary antibiotic usage. So just in case antibiotic is prescribed, even though that's reducing in some areas, it's still a large problem globally. And on the right-hand side, you can see some of the respiratory analytes that Medix Biochemica offers. And the little dots indicate whether we have antibodies or antigens available. And we really run the gamut of respiratory infectious diseases from influenza A, B, RSV, Legionella, strep A, Bordetella pertussis, adenovirus, Mycoplasma pneumoniae, and metapneumovirus. For a little bit of some of the data we've uncovered as we've been conducting a lot of internal testing with all of our clones from different brands. One thing that we've noticed that has come up a few times is that we may identify the best pair in ELISA or fluorescence immunoassay, and sometimes that pair is still the best pair in lateral flow, or in this case, it's the best pair, but in lateral flow, it would be used in the opposite orientation. So V1371 is the catalog code for one antibody, and in fluorescent immunoassay, it's best used as the conjugate, but in lateral flow, it's best used as the capture. So this is something to keep in mind. And this same phenomenon is something we noticed with our influenza B antibodies. However, with our RSV antibodies, we didn't see this, and the top antibody pairs stayed consistent orientation for fluorescent immunoassay and lateral flow. So you really don't know, and you need to have this information provided to you, and we can do that, and Richard can do that at Lateral Dx. So that the data that I just mentioned to you is available at medixbiochemica.com at our resource page, and there's also plenty of more information for you to discover. In the middle of the slide, you can see a poster presentation for some of that RSV data that our team presented at ECCMID this year. And there's other poster presentations, white papers, and tech notes that are just full of information that are helpful for you. Another issue facing the globe are sexually transmitted infections and viral hepatitis. And these are issues globally. And over 1 million sexually transmitted infections occur per year worldwide. And many of these infections are managed but not curable. Over 300 million people are estimated to have chronic hepatitis B and C worldwide. And there's over 1 million deaths from those chronic infections worldwide every year. This is just a quick slide to share the analytes and the antibodies and antigens that we have as solutions for you for test development for those areas. And we also will have an upcoming HIV P24 antibody pair available very soon for pre-launch samples. And our data so far is very promising, and this looks to be a very sensitive pair, ideal for 4th generation tests. Another grouping is vector-borne disease and gastrointestinal infections. And this is just a quick snapshot of our product selections here. And for your assay verification and validation, we also offer a number of different biological samples for you to use. We have base matrices, which are large scale process plasma and serum. And these can be highly customized. So please get in touch with our team. These are control-based matrices, positive or negative disease state plasma, and a number of other base matrices that would be of interest to you. We also have a category of biologicals and biospecimens that can be provided at a smaller scale in some cases. There's urine that can be provided, and cerebral spinal fluid, and various swab samples that can also be customized according to your needs for categories such as age or gender, or diet. So as Anthony mentioned, we're celebrating our 40th year anniversary of providing our products to the IVD community. And we're going to keep continuing to supply our quality products and with a reliable supply, keep in close touch with our customers, and innovate scientifically with our R&D teams that are spread across the globe, and continue providing a comprehensive portfolio for you so that you can develop the tests that the world needs. And currently we are offering a select infectious disease products free of charge sampling, so that you can try products that you maybe haven't tested before from us. So thank you.
Excellent. Well, thank you, Amy. I will take control over. Let's see. All right. Well, thank you, Richard, and thank you, Amy, both. Thanks for the important information for such a timely topic. Right now, we'll get into the Q&A session. But first, I have one more ask of you guys, and that's just for your feedback. We want to provide the best, most relevant information for you guys and make these webinars of the most value for you. So we encourage your feedback. And in just a few minutes, I'll also provide a link for you to click through in the chat. So be on the lookout for that. And thank you all for the questions that you've already provided. I know we've got just a few more minutes, so I'll work through just a couple of them. And let's see. First up, we have, I think this one is mostly for you, Richard, but although you both mentioned it. And that's, what is the right time to test biospecimens in your assay development process?
Okay, yeah, Anthony, I can make a stab at that one. So as I was saying in my presentation, it's really important that we do include biospecimens in the development process. Where to start using them is a question of debate, I would say. We usually start with either a buffer matrix or a stripped serum or a kind of base matrix, artificial matrix, to do some of the antibody screening. But we'll quickly confirm those results of anything that we select with analytes spiked into matrix or actual characterized samples. So I would recommend that you have a small panel in your feasibility study of maybe 10 samples that are well characterized and go and cover the range that you want to cover in your assay development. Then as you move through the process, you add more and more samples to each kind of testing point that you're doing. They are expensive, you know, but they are very useful and can save a lot of time if you have a reasonable number when you're starting. So short answer, probably about 10 within the feasibility phase.
Thank you. I think this one might be for both of you, but somebody said, how many reagent lots should be tested to ensure robustness, and what exactly qualifies as a quote-unquote lot?
Amy, I'm okay taking the first part if you want to maybe answer the second part.
Okay.
So yeah, usually people would say 3 lots of the critical raw materials would be what you would want to look at during your development process. So yeah, I would say 3 is good. It's a good number. More than that, obviously, would be better. But I think 3 is the accepted industry standard for lots of material.
That's been my experience too, that 3 lots is the sweet spot. And for what a lot is, what a lot means, or a batch should be separate purification of material. And where you're obtaining these materials, there should be stringent traceability so that these lots can be tracked back to their source, whether it's a particular supernatant or ascites or recombinant expression batch.
Yeah, Amy, I would agree, because maybe different manufacturers call a lot something different, and you might be thinking you're evaluating three separate lots that have come from three separate starting points, but it might be three separate purification runs. That might be okay. It's the knowledge and it's the information from your provider that you're really looking for, so that you know what you're testing when you do those evaluations. That,
Mm.
to me, is the important part.
Mm.
Okay, great. Thank you both. Do you offer custom development solutions to meet customer needs, unique customer needs?
Yes, yes. At Medix Biochemica, we do, and we're very happy to co-develop or partner with any customer for recombinant antigens, native antigens, antibodies. Richard?
Yeah, and obviously every project we do here at Lateral Dx, it is a custom product. There isn't really anything off the shelf, so we're really tailoring that development to what the customer needs and what the final test performance needs to be. We're going through similar processes, but yes, generally it's customized to the customer.
Okay. Time for maybe just two more. So first one being, with so much attention on emerging outbreaks, how does your team respond to these urgent situations?
Hmm. For at Medix Biochemica, for all of our production sites, we're really built for IVD customers. So we're able to scale up quickly when the need arises. Obviously, it's nice to have forecasts, but we can easily scale up and have on many occasions when the need arises, when we get that call from our customer.
Yeah, so if you imagine that there's a new infectious disease, or one that's existed before, maybe makes a jump or moves to a different location, and there's that clamor to develop a new test. It's how quickly can you get through that process. So you've got to be able to find the reagents, the antibodies. Generally, they may well be available, or they might have to be developed. If they have to be developed, that's a longer process. But as we're moving into kind of recombinant technology in that field, that's becoming quicker and quicker. Then when it comes to doing, proof of concepts, we usually do those in about 4 to 6 weeks. And from that, you can have a very good idea of your test's performance and how it's going to work. So, I think in a good 3 months, you can have a nice feasibility test that you can be taking out for evaluation on available samples, or user trials. And that kind of time scale kind of works, I would think.
Okay, thank you both.
Anthony, I
Sure.
should also add that we also have a large pipeline in our R&D teams. So if there's an outbreak and you don't see a catalog product on our website, it's very possible it's something that is in development right now. So ask us, or we could also consider a custom project with you.
Cool.
Yeah, and Amy, we've done that. Come to you saying there's this requirement, and you said, yeah, it's in the pipeline. Do you want to have this kind of pre-sample, trial it out? So yeah, I would encourage people to just ask, because you don't know. There's lots of stuff getting worked on, isn't there, Amy, with that might be at a stage that is ready for evaluation.
Excellent. Thank you both. And this is a fun one, and I think this will be our last one, and I'm interested as myself for both of your answers. And somebody asked, you're working with some pretty scary stuff. Has that changed how you go about things, either at work or in your everyday life?
Um, I, I guess for myself, I'm still doing some COVID testing. Using hand sanitizer much more than I ever did prior to COVID. But as a microbiologist, I think I've always been aware of what's around us and had healthy respect, but also I'm not paranoid either or nervous.
Yeah, I think I'd maybe similar, Amy. I think it raises your awareness. So some of those risky things, it's very unlikely I'm going to get in a hot tub now at a holiday rental unless I really know it's properly been cleaned and tested. Um, I don't see myself taking too many more kind of precautions when traveling, but maybe I should. And I do use lateral flow tests a lot. You can do a lot of testing yourself, but I think you've got to have a little bit of knowledge about what the result means and that might not be for everybody. I've also, stopped working in the lab, and that's probably the safest thing for me, actually, for for not limiting my exposure to these agents.
Well, thank you both. Yeah, a good healthy respect is, but don't let it rule your life. I suppose that's a good takeaway. Well, thank you both. Thanks, everybody, for your time, for the questions, for the feedback. Again, if any other questions or comments, or to speak out to reach out to Richard or Amy, please reach out to us at info@medixbiochemica.com. And with that, one more thank you, and we'll see you at the next one. Bye, everybody.
Thanks.